Nuclear membrane is again a double one. The inner one is called the inner membrane, the outer one is called the outer membrane, and there's a gap in between which can be called as perinuclear space or intermembranal space. Can you make out there are some places where gap is present? These gap-like portions are called as nuclear pore. Now why are these pores important? Because a cell has to have communication going on with the rest of the cell components. So the cytoplasm is always in a continuous dialogue with the nucleus. Things are entering inside the nucleus, things are moving outside the nucleus. So all these things are controlled by the nuclear pore. Now another thing that you can see the membrane is in contact with the RER. This is RER. So you can see there are ribosomes present even on the surface of the nucleus or nuclear membrane. Outer surface is in continuation with the RER. That means the things which are formed in the RER can enter inside the nucleus, or if a nucleus synthesizes something important, it can shift it outside the cell even through the ER. That's why, if you remember, I gave you a word endomembrane system, because I told you that the ER basically plays an important role in transportation in and out of a cell. So this is the another diagram which is given in NCERT for the structure of nucleus. So the liquid present inside, just like cytoplasm, the liquid present inside the background is called as the nucleoplasm. And then there is another small body present just inside the nucleus, which is like a smaller nucleus, is called as nucleolus. Then there are holes in the membrane, which is called as nuclear pore, and then there is a double membrane envelope, which is called as the nuclear membrane. So what are the various parts of a nuclear envelope? So the first, what are the various parts of a nucleus? The first part is the nuclear envelope, which is also called as the karyotheca. It's all mentioned in the notes, karyotheca and the nucleolema. The outer membrane and inner membrane. So outer membrane is also called as ectokaryotheca. Ecto means outside, karyo means nucleus, theca means the wall of the cavity. And the inner membrane, IM, is also called as endokaryotheca. Now, mistake in the NCERT otherwise. Endokaryotheca is always smooth because it does not carry ribosomes attached on its surface. But the ectokaryotheca may generally appear rough in nature because it has ribosomes attached to its outer surface, as well as it is connected or is in clear connection with the RER. The gap between the two, the sizes, first of all, the size of the outer membrane and the inner membrane is 60 to 90 angstrom in thickness. And there is a gap in between two, which is called as the intermembranal space or perinuclear space, which is 100 to 500 angstrom in thickness. Remember, this space is in connection with the lumen of the RER. From that NCERT diagram, it has been made clear by me. So the nuclear membrane is done. Now the next part is nuclear pore. I have already told you, it is selectively permeable type of membrane. It won't allow everything to come inside. It is selective in nature. Now the next part of the nucleus is the nucleoplasm. The fluid content present inside the nucleus is called as nucleoplasm, also called as karyoplasm or the nuclear sap. Now it carries the chemicals, it carries the substances. The background substances are called as the ground substances and chemicals. Now which chemicals are there apart from water, because water is the universal thing in every cell. Apart from water, it carries good number of chemicals, and those chemicals may include minerals, enzymes, proteins, nucleotides. All are involved in the metabolic reactions. So nucleoplasm carries something inside it which is called as the skeleton. Now we all know that a cell has to maintain its shape, size and all. Same way, a nucleus, being the biggest portion of the biggest component of a eukaryotic cell, also has to maintain its particular shape and size, because there is a liquid inside. That liquid may cause change in the shape, and that can cause problem with the DNA inside. That's why a nucleus has to maintain its shape. So that means there should be a skeleton. That skeleton is called as the nuclear skeleton or the nuclear matrix, and it is made up of intermediate filaments. Intermediate filaments are of two types, cytoplasmic and nuclear. So which one is present in it? Nuclear intermediate filament. These filaments are made up of protein. We'll be talking about that on the next sheet, because there is a topic of skeleton also in this chapter. Now the next part of the nucleus is chromatin. What is chromatin? We all know that DNA and protein. DNA full form is deoxyribonucleic acid. So it carries a deoxy sugar, a pentose sugar or ribose sugar, and this sugar joins with the nitrogen bases. In the last class I told you A, T, G, C. These are types of nitrogen bases, and then there is a phosphate group. These three things, which things? A pentose sugar, deoxyribose sugar is the pentose sugar, 5-carbon sugar. The sugar joins with the nitrogen bases and join with the phosphate group to form something which is the building block for a DNA. And when many of these join together, it forms a long chain called as DNA. Now this long DNA cannot be inside a very small nucleus, inserted easily. That's why there is requirement of packaging. It has to be packed or arranged nicely. This arrangement is done with the help of positively charged proteins. Reason: DNA is negatively charged. DNA carries phosphate, and phosphate is negative in charge. So this negative charge compound can be stabilized with the help of a positively charged protein, and those proteins are called as histone proteins. In the next chapter, DNA topic is coming in biomolecules. These topics will be made clear to you. So DNA and protein, they join nicely to form a compact structure which looks like a beads on a string. It is something like lots of beads arranged on a string or a thread. So that structure is called as chromatin. So this structure was explained by Fleming in 1879, and he used basic dyes to do this observation, and he discovered that chromatin reticulum is present. There is a network. Reticulum means network of chromatin is present inside the nucleoplasm. And today we know that chromatin is of two types, heterochromatin and euchromatin. That means some portions